body's natural way of doing it. So the cardiac differentiation model, this is what we've been doing over the past 2 years at Oxnard College. We've gotten rather good at it. So we've been able to control the differentiation of these stem cells into the beating cardiac adherent tissue in the following manner. And again, this is where we got the example of the applesauce video that I showed. So we just took mouse stem cells, we differentiated them into cardiomyocytes using a 1% DMSO solution in a standard alpha MEM solution in non-tissue culture plates. So what we do, the interesting fact for this one is we do use the 1% DMSO, which is a little different. And we also only change the medium twice a week. Due to the time constraints we have in our lab, we only are able to get in there really 2 days a week. We don't have a lot of resources. And we're working on that. But we only change the media twice a week, so that's worth noting. So I'm going to show some different types of beating that we get using this technique. And the two ones are the twin beating or twin beating centers and the edge effect. And the images that you're going to see are from our microscope. We attach the camera to it, which projects to an HD TV, and then we take pictures of that. So again, we need a little better. So this is an example of edge effect beating. So when, in this, what's going on, this is the edge of the structure, and you can notice that it is beating very vigorously around the edge. What's happening is those cells are actually under tension. And as the cells come under tension, they begin to lift off the plate, and they start to form these parallel beating centers. And those are actually good because that's what we want later on in heart tissue. And so we really paid attention to a lot of these edge beating effects. And this next one is another example of a 2-beat video in which you will actually see there are 2 beating centers here, which are attached there, there, and there. And a possible area of study that we have later is to examine those areas of attachment, because that could be where possible pacemaker cells are located. So that's kind of a future direction. So we performed a lot of experiments over the summer to get to the experiment I'm going to talk about in the end. I'm going to have to skip over some of these because due to time, but one I do need to talk about is adherence experiments. So what we needed to know is, can we grow the cardiac tissue that beats when it doesn't adhere to a tissue culture plate? So what we did was we did it on our non-tissue coated plates, which we know works, and also ultra-low binding plates, which does not allow for adherence, and so the cells will grow off of the plate. And what we found is we didn't get any beating cells in those ultra-low plates. So we concluded that we do need adherence to a substrate in order to get the beating. Rare earth magnets, we did this one real quick. We didn't really notice any different with magnetic fields. Extra Wnt factors and direct differentiation experiment. I'm going to skip over those. What we did was we tried to add some different either Wnt factors or these 4 drugs to see if they got any better results than how we're doing our cells. What we noticed is the cells did not beat nearly as long, if at all, and some of that might be the application we put them on, so we're going to redo these ones later on in the future. So we decided to continue on with the experiment that we've been using with our 1% DMSO media. So fibrin overlay experiment. This is where things start to get interesting for us, because now what we did was we took cells that beat, okay, and we actually added fibrin, which is just fibrinogen and thrombin, over the top of it, and we wanted to see what would happen. And what we actually got was, on that the layer that was on top, is we actually got beating cardiac tissue in rather large structures that that we were able to that had multiple uncoordinated beating centers. So I'm going to show you a couple examples of this. We call this one iguana. Before I play, I just want to let you know this is the overlying fibrin. These are the cells that it's growing over. Okay, as you can tell with this video. You see very vigorous, well, if I can get it to play, there we go. There's very vigorous beating going on in this overlaying fibrin structure, and it's all throughout the fibrin that grew on top of the pre-existing cells. And the structure was so large in this next video that we could actually see it with the naked eye. So this is actually taken, we could see movement with our naked eye looking down at the plate. So on this plate, you can actually see that was beating. That's taken from a dissection microscope, not a regular microscope that we use And this is an example of gear.
Okay, this is the structure that we found at 5 weeks.
So as you can see with gear, this is the overlaying fiber and structure. There is beading going on, and it's creating a lot of pull. You can see the tension, and you can see it moving again. And this would be a good way to actually build functional structures later on. We're going to play with this one a little more too. So finally, before I can start talking about the experiment I need to talk about, we had to decide what the 3D cast was going to do. First, we made these little die-like squares. We ultimately didn't use these because the gelatin kept leaking out the bottom, and the oxygen diffusion was very poor. So we made these boats, and these boats basically are bathtub shaped. We edited them to get them to fit inside the well, and then we decided to go on with that. So the goal of our final experiment is to create a novel and inexpensive fibrin gelatin stem cell matrix that we can then add to a 3D mold in order to create large beating cardiac structure. I'm going to have to go over this pretty quick because I'm running out of time. So first we built it on the 3D printer. Again, this is an example of our 3D printer in motion. We added the Matrigel substitute to the boats, and it consisted of the bloom gelatin in the alpha MEM media, fibrinogen, mouse cell, embryonic bodies, placed it in the boats, added thrombin on top. You can see a little polymerization going on there in the boats. Let it sit 5 minutes. We removed the gelatin into a cell strainer, and then that allowed us to be able to transfer it over and over again to different media at our media change. We had 3 different types of media. The control media, which is on the far left, that was just our standard alpha MEM mixture. Media 1 had DMSO in it, and media 2 had both DMSO and gelatin. And we changed the media again twice a week, and this is where we got all our observations and our documentation done. As you can see, so these are some of the end results of this. So we did get rather large macrostructures growing in all of the media. So this is control 1 and 2. These range anywhere from 2 to 3.5 centimeters long and almost just as wide. Let's see, there was beading going on in all of them, but it was more vigorous in media 1 and media 2 than the control. One downside to it, though, was that it was very hard to be able to view anything inside the large macrostructure. But you can see beading. This is more from the edge, but you can see that there is beading tissue throughout that macrostructure. And also, what an interesting thing we found was the cardiac macrostructures were actually able to use the cell strainer itself as a substrate to be able to keep growing. So you can see it starting to grow around the cell strainer itself. So that's all I got for time. So I figured I'd end on saying we have a little heart-shaped heart that beats. I figured that would get some kind of response. So we have some very, very cool stuff going on. We are going to be doing this more in the future. We want to work on the different sizes of getting it down to where we can actually see what's going in the macrostructure. Lots of different possibilities with that. So do you have any questions?
Woo!
Very exciting project. Guys, do you have any questions?
Blown away.
Yes.
Yes.
When you treated your cells with Wnt, did you use a conditioned media?
We used C59 and XAB. So what happened with Wnt was we wanted to see if we could get more beading using these molecules than we did with our 1% DMSO. We did not. We actually found that we got less beading. They did get beading, but it wasn't nearly, they didn't go for as long. And it actually affected them a little bit negatively. But we're going to go back and try this again. Maybe instead of like leaving it on as a continuous application, apply it and extract it, see if that has any better results. But again, this was kind of our first try. Any other questions? Yes?
So the reagent used was gelatin and
It was, yeah, it was gelatin. It was, what we did was we made, it was 3% Bloom 75 gelatin that instead of mixing it with water, we mix it in with that alpha amino medium.
Okay.
And then we added fibrinogen to that, along with the EVs that we just unfroze. We added that solution to the boat, and then we added the thrombin on top of that to polymerize in the boat. So that's how we created the 3D structure. Because we always see a lot of stuff, on plates beating, but we don't see a whole lot of 3D structures. So that was the goal, is we wanted to be able to get actual beating 3D structures that weren't just stuck on a plate. Because that would have really good applications later on if we could take those beating cells from a patient's own cells and actually apply them to their own body. That would hopefully in the future eliminate a lot of these really invasive procedures.
Transplants.
Thank you very much.